Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper part C questions of shift 1. This was conducted in July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR been answered, you can go to my channel Omni Bioentrance in the playlist C for CSIR Net and you will get it there. Coming to the first question. The following gel pattern are the DNA markers observed in parents P1 and P2. These are P1, P2, P1, P2, P1, P2. F1 from a cross between them. Okay, this is the F1 obtained after crossing P1 and P2. Okay, this one. And from the cross between them, and double haploid progeny, panel A. So this is of panel double haploid progeny. These bands are of double haploid progeny, or F2 progeny derived by selfing F1 panel B and C. So B and C is having F2 progeny, not double haploids. Okay. How do you get these double haploids? So double haploids is a genotype formed when plants are developed from haploid cells which have undergone chromosome doubling. So from the diploid plant, you isolate the pollen grains or the egg cells, and then you so they will be homozygous for all the genes because they are haploid. And then you add some chemicals like colchicine for chromosome doubling of these haploid pollen grain and egg cell, and you develop plants from them. So it is called as double haploid plants. Based on the pattern observed in the double haploid or F2 progeny, identify which of the patterns A to C are based on DNA markers that are allelic. So DNA markers that are allelic are naturally your codominant markers. That is, they should have both alleles, that is capital A and small a in them. Okay, that means they should have both P1 and P2 as per this question. So this is P1 parent, P2 parent. F1 is having both the bands of P1 and P2. Then a double haploid, since they are derived from haploid cells by chromosome doubling. They will have P1 band, and the other plant will show the P2 band. So that is showing all the bands. And it is showing a consistent banding pattern. So that is a case of allelic DNA markers. Now in F2 progeny, here in the panel B, if you see F1 and F2 showing both the P1 and P2 bands. F2 progenies are also showing P1 and P2 bands. This is showing P1 band. This lane is showing P1 and P2. This lane is showing only P2 band. So F2 progeny in panel B is also having allelic markers. Now if you see C, you can see that F1 is showing P1 P2 band. This lane is also showing P1. This lane is also showing P1 P2 band, but this lane doesn't show any bands. So this might include non-allelic DNA markers. Okay, C. So they have asked here for DNA markers that are allelic. So your answer should be A and B. Answer is option 4. Then next one. A KDL sequence is added at the C terminus of a secreted glycoprotein X. Okay, now what is this KDL sequence? So KDL sequence, it prevents proteins from being secreted from the endoplasmic reticulum and helps these proteins to return back to endoplasmic reticulum by chance if they are exported. That means if they go to Golgi complex, then they can be exported back to endoplasmic reticulum because of the presence of this KDL sequence. Now what this experiment is doing is they have added KDL sequence to the C terminus of a secreted glycoprotein X. So formerly this glycoprotein X, it was secreted. Now KDL sequence is added to it, so naturally it will go to endoplasmic reticulum. Then 500 amino acid residues having no site for end-linked glycosylation and expressed from an inducible promoter. Okay. Following 10, 20 and 60 minutes of induction. 10, 20, 60. 10, 20, 60 minutes of induction. Endoplasmic reticulum are purified and probed for newly synthesized glycoprotein XKDEL. The immunoblot obtained is as shown below. So this is the immunoblot. Now at 10 minutes you can see it is 55 kDa. But at 20 and 60 minutes you can see at 20 minutes there is a band of high molecular weight. So that means there is some modification in KDL. No, not KDL, but modification in the glycoprotein X. That is why you got higher bands, whereas at 10 minutes it didn't undergo any modification. That is why you didn't get any higher molecular weight band. Okay, now read the statements. Which of the following statement is most likely explanation for the presence of the high molecular weight bands in lane 20 and 60 minutes? The signal sequence is not removed from some of the glycoprotein X KDL molecules. Now if KDL molecule was removed, then the protein will be secreted out of the cell. It will not be going to the endoplasmic reticulum. Okay, now they have mentioned that endoplasmic reticulum are purified and probed for newly synthesized glycoprotein X KDL. How will you get that answer? If it was if KDL sequence was removed and it got secreted outside of the cell. Okay. Now next one. Glycoprotein X KDL become modified in the endoplasmic reticulum after protein synthesis is completed. If that was the case, then at 10 minutes also you should have got a higher band, higher molecular weight band, that is at 60 kDa. But that is not happening. The glycoprotein X KDL molecules are modified in the Golgi prior to endoplasmic reticulum retrieval. That is correct. Why? Because you got higher molecular weight band at 20 and 60. So first maximum was at 60. Higher molecular weight band at 20 it was less, and some 55 kDa band also was observed. Now why did I say that third is the right answer? Because they can enter the Golgi complex first, then undergo modification there, and I told you because of the KDL sequence they can be transported back to endoplasmic reticulum via what is called as retrograde transport. Okay, so they could have got modified in the Golgi and then they would have entered into the endoplasmic reticulum by retrograde transport. So 3 is the right answer. The quality control mechanism in the endoplasmic reticulum recognizes a pool of glycoprotein XKDL as being aberrant and targets it for degradation. If it was degraded, if suppose the KDL was degraded, XKDL was glycoprotein XKDEL was degraded, then you would not have got high molecular weight bands, you would have got still lower, very much lower. So fourth is not the correct option. So answer is option 3. Given below are a few statements on the concepts of molecular breeding. Correlation between which are the correct statements? Okay. Correlation between quantitative traits can be because of pleiotropic effects of the same gene and or genetic linkage of genes associated with the traits. That statement is correct. Now what is pleiotropy? It is the expression of different phenotypes by a single gene. And quantitative traits, it is controlled by polygenes, that is controlled by multiple genes. So genetic linkage, it refers to genes located close together on the chromosome are inherited together, and these both mechanism can cause correlation between quantitative trait is correct. So A is correct. In recombinant inbred lines population, genetic segregation of both dominant and co-dominant markers occur in a 1:1 ratio. So recombinant inbred lines or RIL, they are developed through repeated selfing of the F1 hybrids. And they ultimately display homozygous genotypes which segregate in the 1:1 ratio. So B is also correct. Near isogenic lines can be produced by repeated backcrossing of the F1 to a recurrent parent. That is correct. SNPs are dominant markers. They are not dominant. They are co-dominant markers. So A, B, C is the right answer. Answer is option 3. Then next one. The RAN GTPase imposes directionality on transport through nuclear pore complexes. Like other GTPases, RAN is a molecular switch that can exist in two conformational states depending on whether bound to GDP or GTP. So there is RAN GDP as well as RAN GTP. Now possible reason for compartmentalization of RAN GTP accumulation is, which are the correct statements? So RAN GAP is enriched in the nucleus. No, it is in the cytosol where RAN GAP is found. RAN GAP is localized in the cytosol. That's correct. RAN GEF is enriched in the nucleus. That is also correct. So B and C are correct. Answer is option 3. Then next one. A cancer clinic is treating 4 unrelated patients suffering from chronic myelogenous leukemia. Okay, Philadelphia chromosome. Remember, a researcher sequences the Philadelphia chromosome from the leukemic cells of these patients and make the following statements. Now, what is CML? It is translocation between the long arms of chromosome number 9 and chromosome number 22. And the translocation, it fuses the BCR gene on chromosome 22 with the ABL1 gene from chromosome 9. That is what is happening in Philadelphia chromosome. Okay, now first statement. The DNA sequence was identical in the translocation, breakage and rejoining sections in all leukemic patients in all 4 patients. That is incorrect. So wherever A comes, it is not correct. So 1 and 4, no. Because the break point and sequence in the Philadelphia chromosome can vary between different patients. That is, wherever the chromosome break and rejoin, that is your TBR section, it can differ among patients. But it will be the same in one single patient. If one single patient is having a breakage at position A, it will be position A in all the chromosomes in that single patient, but not in other patients. In patient 2, it can be at position B. In patient 3, it can be at C, like that. Now next one. The DNA sequence was identical in all leukemic cells from patient 1. Yes. But every patient had different TBR sequence. I explained that just now. So B is correct. All patients have translocation between long arm of chromosome 9 and 22. Yes. That is correct. That is Philadelphia chromosome. All patients have translocation between long arm of chromosome 9 and short arm of chromosome 22. That is also not correct. So wherever D comes, that is wrong. So third option is wrong. Your right answer is B and C. Answer is option 2. Now given below are protein domains and their binding specificities. SH2 domain. SH2 domain, phosphorylated tyrosine residue on receptors. SH3 domain, short proline rich amino acid sequence on proteins. pH domain, charged head groups of specific phosphoinositides on plasma membrane. PTB domain, it is phosphorylated tyrosine residue on receptors. So the answer here is option 4, A1, B3, C2, D1. Now next one. Some features mentioned below are important for segregation of homologous chromosome in meiosis 1. Synaptonymal complex formation between homologous chromosome. Yes, that's correct. Happens during prophase 1 of meiosis. So A is there in all the options. Degradation of cohesines at the chromosome. Yes. During meiosis 1, cohesines which hold the sister chromatids together, they get degraded at the arms of the chromosome, but this degradation doesn't happen at the region where the chromosomes are connected at the centromere. Okay, this is for ensuring proper segregation. So B is also correct. Retention of cohesines at the centromere. Yes, that I told you just now. Biorientation of kinetochores of sister chromatid. Biorientation happens during metaphase in mitosis and metaphase in 2 in meiosis 2, not in meiosis 1. So D is also not, D is not correct. So the answer is A, B, C only. Then next one. Given below are some components that could potentially influence membrane fluidity. Monomeric G proteins. They are involved in intracellular signaling and they do not play any role in fluidity. So the 1 is not the answer. So 1 and 2 gone from the options. Peripheral membrane proteins. They are bound to the surface of the membrane and are not embedded in the lipid bilayer. So therefore they do not affect fluidity. So wherever 2 comes, that is also gone. So what is left out? 3 is the answer. Sphingolipids, they influence membrane fluidity. They are involved in lipid raft formation. Then phospholipids, side chain saturation. Yes. Saturated fatty acids, they have no double bonds. They pack tightly. They reduce fluidity. Whereas if it is unsaturated fatty acid, they have double bonds. They create kinks, okay, KINKS, and prevent tight packaging and increase membrane fluidity. Then cholesterol, at high temperatures it decreases fluidity, at low temperatures it increases fluidity. So 3, 4 and 5 is the answer. Then phylogenetic trees are used to examine relatedness among different population species genera. Yes. Similarity in characters among different population species genera. Yes. Common ancestry among different population species genera. Yes. So A, B, C are correct. 3 is the answer. Functional significance of traits is not taken into consideration in phylogenetic trees. So D is not the answer. A, B, C is the answer. Then given below are statements on concepts of genetics. Which of the following option represent the most appropriate sequence of terms to fill in the blanks in the above statements? The degree to which a particular gene is expressed in a phenotype is called expressivity. That is there only in option 1. So option 1 is your answer. A heritable change in a gene expression that does not result from a change in the nucleotide sequence of the genome is called epigenetic change. It is heritable, but it doesn't cause any changes in the nucleotide sequence of the DNA. So it is epigenetic. The frequency with which a dominant or homozygous recessive gene is phenotypically expressed within a population is called penetrance. An allele that results in the death of an organism that is homozygous for the allele, that is recessive lethal. So first one is the answer. Then next one. Which of the following options represent a classical Hoogstein base pairing? So this is the normal Watson base pairing. Anti-A, anti-T, anti-G, anti-C. Now if this A base pairs with anti-T, that is when the base A's different atoms are involved in base pairing with anti-T, then A is in syn conformation and T remains the same, anti-T just like Watson-Crick model. So this is called as Hoogstein base pairing. Similarly, this purine also, it can base pair with anti-T at different positions, not the normal Watson-Crick base pair positions. So here it is double bond, here it is triple bond. See the difference? So it is syn G with anti-C. So here if you see the options, it is syn A base paired with anti-T. Answer is option 3. Then anti-G base paired with anti-U, anti-G base paired with anti-C, anti-A base paired with anti-T, all that are not correct. Then next one. Given below are few statements regarding the rate of glycolysis, gluconeogenesis and glycogen metabolism. Increased blood glucose would decrease gluconeogenesis, of course, and increase glycogen synthesis, that's 100% correct. So wherever A is there, that is correct. So 2 is not correct. 4 is also not correct. We will see 1 and 3. B. Increased level of fructose 1,6 biphosphate inhibits glycolysis. That is incorrect because fructose 1,6 biphosphate, it is an intermediate of glycolysis and it stimulates the process of glycolysis, not inhibit it. Anyway, B is wrong here in 2 and 4. So B is wrong. Increased blood glucagon inhibits glycogen synthesis and stimulates glycogen breakdown. That's correct. So glucagon is released when blood glucose are low and it signals the liver to break down glycogen into glucose and decrease glycogen synthesis, thereby increasing blood glucose level. So C is also correct. So you got your answer, which is A, third one, A and C. D. Okay. Increase AMP level inhibit glycolysis and stimulate gluconeogenesis. If it is high AMP levels, it means the cell is having low energy. So that means glycolysis is getting activated to produce ATP. So D is incorrect. Then next one. A complete retrotransposon was cloned under the entire galactose-inducible promoter. This is the GAL promoter, this is the retrotransposon. The construct as shown below was inserted in the yeast genome to study transposition events. Now what is GAL promoter? It is inducible in the presence of galactose in the media, leading to expression of the retrotransposon. Okay. Now retrotransposon, what is that? It is a transposon that is inserted into new locations, jumping via an RNA intermediate. Now read the statements. Cells grown in the presence of glucose or galactose lead to increase in copy number of transposon, not in the presence of glucose. So A is not correct. Galactose, okay, but not glucose. So A is not correct. The transposed copies will be the same as the construct inserted in the genome. No, the transposed copies can never be same just like the construct inserted in the genome. Because when transposons move, they often integrate into the genome at random sites. And these transposons, once transposed, they can still undergo transposition events. So the copies of transposed will not be same. Then the transposed copies cannot transpose further. They can transpose further. I told you just now. Then the transposed copies will not respond to either glucose or galactose in the media. Yes, that's correct, because they have told transposed copies. They will not be under the control of gal promoter. Okay, so they will not respond to glucose or galactose in the media. They are at an entirely different position on the genome. They transpose randomly in the genome, but they don't carry the gal promoter while transposing. So they will not respond to either glucose or galactose in the media. Assuming that the hypothesis is correct, choose the correct statement. So D only is the answer. Answer is option 4. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CSIR being answered, you can go to my channel Omni Bio Entrance. See the playlist. In the playlist, you can search for CSIR Net JRF. There you will find the previous question papers. So thank you for watching.